Hi, welcome to this lecture on mathematical methods for biologists. We have been discussing statistics and we learned why do we need and when do we use mean or average and standard deviation and what does it imply. In this lecture we will discuss distribution. So, the title is frequency distribution and probability distribution, and the question we will answer is how do we plot the data set as a distribution. So, this is a thing that we would answer in this. We given a data set, how do we plot it as a distribution. Now, while we discuss this, there is an important thing that one should remember, this idea of an ensemble, or a population. So, this is something that this word very often you might what is an ensemble? Typically, imagine that you have an experiment in which you might be looking at many cells, for example, under a microscope. So, all the cells are exactly under the same condition, same temperature, same concentration of nutrients and all that, but they are a population, they are not, they are individual cells. So, this is, let us think of, so you have a petri dish or you have a culture, you have many cells in this. So, you have all of this might have different size a little bit, it might have different shape, but these are all same cell type. So, these are all same cell type, same condition, same cell type. So, let us say they are all yeast cells or they are all HeLa cells or same cell type, same condition. So, the things like temperature, pressure and all that, this is all fixed, and what we have fixed is number of cells is given here, and the nutrients, the concentrations, like the concentration of nutrients, concentration of chemicals, nutrients, whatever in this we have, which is all essentially the same for all the cells. So, concentration of nutrients, let me write, they are all essentially seeing the same condition. So, you have, so this you would call an ensemble of cells or a population. So, these two words would be very often used. You have a population or an ensemble. So, this ensemble of cells, and in such set of cells you can do various measurements for this is. So, you could measure, for example, its size, like you could measure the area of each of these cells, and the area of this would be different from the area of this. So, they would all get, you will get different numbers. So, you can get a set of numbers, let us say you are measuring area, so you would get a set of numbers like, so area. So, you would get some numbers in micrometer square. So, let us say 100 micrometers square or 78 micrometers square or 120 micrometers square. So, these are typical examples and you can get many numbers. So, if you have 100 cells, the more the cells, more numbers you can get. So, area is some example I said. You could measure, for example, gene expression. So, there is a fluorescence and you can measure the fluorescence, for example. So, different cells you can measure the fluorescence, like you would get some value in some units. So, value 1, so you would get x1, x2, x3 and many values, like if there are 100 cells you would get x100. So, this is the amount of gene expression. This could be in micromolar or nanomolar or whatever be the unit, but you would get some concentration of proteins expressed in these cells. You would want to measure some other quantity. It need not be cells always. It could be you have a population of, you have a group of people for whom you would want to measure, for example, the vitamin D, like people living in a particular city. They are in the same city under say similar conditions, and you would want to measure their vitamin D level. And something else could be blood pressure, and you would get many different values. So if you have a population of cells or a population of a group of people, or it could be a group of whatever be the organism that you would want to study, you can, any measurement you can do on many individuals, many cells. This is an ensemble, a population, and the data, you would get a large number of measurements. This data, this is one example. Another example could be, you could take one individual and repeatedly measure the same thing. Like you would measure, for example, the blood pressure every day, like, or many times in a day, and that also you will get many measurements. So typically, let us think of population first, and we will come to the measurement as a function of time. So if you have a population of cells like this, or a population of individuals like this, you would measure either its size, gene expression, or you would measure any other quantity like blood pressure or vitamin D level or hemoglobin count. Any quantity that you measure in a population, you will have many numbers like this. So let us say you have numbers between 0 and 100. So you have like many numbers. I am just drawing some example here. So let us say the numbers are 5, 7, 8, 10, 90, 7, 81, 82, 15, 26, 18, 37, 34, 51, 48, 46, 97, and so on and so forth. So you have many numbers. If you have, these are data measured from different, from a population ensemble, you can always calculate the mean. So you can calculate the mean x bar, which we said is sum over I xi divided by n. You can also calculate the standard deviation, which we said is sigma, which is xi minus x bar whole square sum over I divided by n minus 1 and root of this. So this is the two things that we can measure. But you can, the third thing is the distribution, which we briefly mentioned. Distribution is how these numbers are distributed. So from this data, we can make a table. From the data sets we are given, we can ask a question, how many of these numbers are between 0 and 10? So how many of these numbers are between 0 and 10? Let us say, so this is some table that we would create. So let us say 2 of those numbers are between 0 and 10. And how many of them are between 10 and 20? So let us say 5. Then you could ask the question, how many of them are between 20 and 30? So there are many numbers between 20 and 30, 11 numbers out of all those numbers. And how many of them are between 30 and 40? And so there is 15, and so on and so forth. So you could just, finally you will have asked the question, how many numbers are between 90 and 100? So you would get 3. So you got some table like this where you have a range here. So you have a range, and this is frequency. How often numbers fall in this range? So this is frequency. This is range, and this is frequency. So this is frequency, this is the range. What is frequency? How often? How many times? How many times? Or how often numbers fall or appear in a particular range, of our interest. So this is the frequency. How many times numbers fall? So 5 times in this whole population, 5 times the area is between 10 and 20. 11 times the area is between 20 and 30. 15 times the area was between 30 and 40, and so on and so forth. So if you have a table like this, you can plot a histogram. So basically this versus this, we can make a plot. So we would, this is the plot that we would make. So we would have between 0 and 10. So this is between the first range. How many? 3 times. So this is 3. 3 times. So this is 0 to 10 range. And between 10 and 20, the numbers in this range was 5 times. So between 10 to 20 range, so this is 20. In the 10 to 20 range, you had 5 times. 20 to 30 range, you had 7 times. In the 20 to 30 range, the numbers were 7 times. And so on and so forth. You could ask 40 to 50 times, 50 to 60 times, and you would get a distribution like this. So this is 7 and this is 10, and so on and so forth. Some numbers. What is that? So this is called a frequency distribution. So I am going to plot here Fi versus I. So I is the range 1, 2, 3, and this is your frequency for that particular range. So 0 to 10 3 times, 10 to 25 times, 30 to 40, like 10 times, and so on and so forth. So this immediately tells us, if you have a graph like this immediately tells that most of the area fell between 30 and 40. Most of the area fell between 30 and 40. Very few fell between 0 and 10, and very few fell between like whatever the large number. So this is 50, 60, 70. Between 60 and 70, very few fell, and beyond that there was nothing. So there is no number above this, for example. This is one example I am plotting. This tells us how numbers are distributed. How many numbers are between 0 and 10? How many numbers are between 20 and 30? So this graph will tell us how the numbers are distributed in this range. So this is the frequency distribution which most of you know. Now, given this distribution, this range, the smaller the range, of course, the better it would be. The more data we have, we could take the smaller range, and once you have this number, what we can calculate. So let us have this distribution once more. So let us say so the range, we could even take, so let me take some measurement which comes between 0 and 10. So the frequency is f. So let us take here whatever between 0 and 0.1. So the range would be 0 to 1, 1 to 2, 2 to 3, 3 to 4, 4 to 5, 5 to 6, 6 to 7, 7 to 8, 9, 10. These are the range, and corresponding to this range, I would have different numbers. So now this I would represent by a number here. I would, for example, represent by the middle value of this. Let me call this 0.5, 1.5, 2.5, 3.5, and so on and so forth, 9.5. So I have, instead of 0 to 1 range, I just put a value 0.5 here. That's what I put here. Instead of 1 to 2, I could represent this by a middle value which is 1.5. This is one way of representing. 2 to 3, I could put a middle value 2.5. So 2.5 which means around 2.5 how many numbers are there within this range 0.5. Around 3.5 how many numbers are there within this range of 3 to 4, which is half 0.5 up or down. So now you could say there are 7 numbers between 0 and 1, there are 10 numbers, there are 15 numbers, there are 20 numbers, and then there will be like 5 numbers, and there will be like 2. So you could get a frequency like this for every range. Fi versus I. Once you have this frequency distribution, let us understand what are the two things that is meaningful. So sum over I Fi, what does that mean? So Fi is the frequency. How many numbers are between 0 and 1? How many numbers are between around 1.5? How many numbers are around 2.5? How many numbers are around 3.5? When I say around, here I precisely mean between 3 and 4. 3.5 plus 0.5 or minus 0.5. In that range of 0.5 around 3.5, how many numbers are there? That is the thing that I can represent, for example, here. Now let us think of a meaningful quantity here, which is sum over I 1 to 10. In this case there are 10 range values. So if there are 10 of them, sum over I Fi, this gives us the total number of measurements we have. If we have 100 cells, sum over I Fi will be 100. If there are 1000 cells, sum over I Fi will be 1000. If there are 1000 individuals, we are measuring blood pressure of 1000 individuals, sum over I Fi will be 1000. So let us say sum over I Fi is 1000. This implies we measured some quantity, let us say vitamin D level of whatever the measurement we do, 1000 individuals. If the whatever be the measurement, this is what it means. So this is first meaningful quantity. Sum over I Fi is 1000, which is the total number. Total number. So there are two numbers here, the range, which is how many. So there is how many, the serial number 1, 2, 3, 1, 2, 3, so on and so forth, up to whatever be the number, 10 or 11. So it will be, let us say 10 here. Then this is, let me call this as my little n. Little n is 10 here, and there is capital N, which is sum over I Fi. So there are two n's here. Little n is this 10, and some capital N sum over I Fi. Now I can divide this whole thing by the capital N. So I can plot I in the x-axis, I versus Fi divided by sum over I Fi. So that is, I, instead of saying between 0 and 1, I had 10 numbers, this is my Fi, and let us say I am doing experiment on 100 individuals, I could divide this by 100. So let us take an example to do this. So let us consider 100 individuals, and we measured the vitamin D level for this 100 individuals, and we got some value. Okay, so let us take an example of measurement of some medical, let us say vitamin D level. So we are going to plot vitamin D level for a population. So we have a population, and the first one is vitamin D level, and this would be in some typical unit that one would get from a lab. So in some unit, typically this could be around 25. Let us say vitamin D level is around 25 units. We would ask the question, how many people have vitamin D level between 0 and 10 units? How many have vitamin D level around 10 and 20 units? How many have vitamin D levels around 20 and 30 units? How many have between 30 and 40 units? So, and you could ask up to 50, let us say how many have between 40 and 50. So, this is the range of vitamin D level in appropriate units, and the frequency F for each of this range would be given by the second column. So, the frequency for 0 to 10, let us say 3 people have between 0 and 10, and let us say 27 people have between 20 and 30, 10 and 20, and let us say 50 people have between 20 and 30. So, this is 77 plus 380, and let us say 15 people have between 30 and 40, and 5 of them have between 40 and 50. So, let us say this is a measurement that we got from a survey from a population. So, that sum of this 5 plus 15, 20, 70, 73, 70 plus 27, 97. So, this is 100. So, this is 100, and there are 1, 2, 3, 4, 5 ranges we have, and we have 100 people. So, we are doing experiment on 100 people. Now, we can divide. So, sum over I Fi is 100. So, sum over I Fi. So, what is this? This is sum over I Fi. Sum over I Fi. This is 5 plus 15 plus 50 plus 27 plus 3 is 100. Now, I can make a new column here, which is F divided by sum over I Fi. In other words, F divided by 100. So, I can divide each of this number by 100. So, what does that mean? So, this means 3 by 100, which is 0.03. 27, this is 0.27. This is 50 divided by 100, which is 0.5. 15 divided by 100, 0.15. And 5 divided by 100, 0.05. So, this column, this thing we would call as probability. This thing we would call as Pi, which we would call as probability. This is for all this purpose of this course, we will define probability in this way. There is some other mathematical concepts gone into this which we would discuss for this particular course, but for the practical purposes, the frequency divided by sum over Fi, F divided by sum over Fi is Pi. This is an important thing to remember. This thing is called a normalization. So, this process that we did, this is a very important thing, normalization, which is basically calculating Pi is equal to Fi divided by the total. So, here in this case I is equal to 1 to 5 Fi. So, this was, in other words, if there are n range, if there is n range, you should calculate Fi divided by sum over I 1 to small n, which is the range Fi. This quantity is the probability. Now, I can, instead of plotting Fi, I could also plot Pi. So, I could plot Pi versus I, just like, so either I could plot Fi versus I, and you would get some graph. Similarly, I could also plot Pi versus I. So, you would get some distribution like this. Similar, it would look very similar here too. So this would look very similar, just that the y-axis numbers have changed. So here, if this was, if whatever be the number here, this would be divided by some constant, so which is the total. So this would look very similar, but the y value is changed. But this is very important to do this normalization, because let me just come to this. Let us come to this and think about how many people have vitamin D level between 20 to 30, and the answer here is 50. And when I say 50 people have vitamin D level between 20 to 30, when I say this number alone does not make much sense unless I know how many total people are there, how many measurements did we do, how many people did we have in our sample. So that unless we know that, the number 50 alone does not make sense. So this normalization is always very important. In some other context, if you think of it, let us say you took some examination, and if I just say that 1000 people got, 1000 people failed. When I say 1000 people failed, it depends on how many people we have. If they have 1 million people and 1000 people failed, that is very different from having 2000 people and out of that 1000 people failed. So this is important to know how many people are there in total and divide that by that. So this normalization process is very, very important in statistics. If you do not normalize, it may not have meaning always. So always is good to plot pi as opposed to fi. Always good to plot pi and not really fi, because normalization is very important. Pi will have more meaningful, pi will have some more meaning in the sense that we could interpret it more accurately, the interpretation will be more accurate. So I urge all of you to plot always pi as much as possible and interpret things from pi. And in the next lecture, we will discuss how to interpret things from the pi. And of course, fi and pi are essentially the same thing, but if you report, is always report pi, and fi can be sometimes the typical value plugged out of fi can be misleading. Therefore, always do plot pi. Okay, now with this, I would summarize. Probability distribution tells us how the numbers are distributed. It's always important to remember what is ensemble population and for what population are we drawing this probability distribution. Think about it, and we will discuss more about this in the coming lectures, but always compute pi and plot pi, which will have meaningful quantity. We can interpret it and compute some meaningful quantities from pi. With this, we will stop this lecture and continue in the next lecture.